The end is here. I can't believe it. So today is one exercise. It's ELISA. And there's two different types of ELISAs that you can do. You can do antibody detection, which is what we're going to do today. Right, so that was the answer to your bonus, right? Antibodies. And so sometimes referred to as the indirect ELISA. Right, so sometimes you'll see that term used for antibody detection. So ELISA actually stands, or ELISA stands, it's an acronym. It stands for Enzyme-Linked Immunosorbent Assay. So we use a special antibody, that's the immuno component, for these types of tests. So an assay is a series of steps. Okay. So again, you have to follow them in series, otherwise it won't come out correctly. So enzyme, of course, it refers to the enzyme that is attached to the antibody. And in this case, it's the secondary antibody. It's going to be the second antibody we use. So as I said, immuno refers to the antibody, which is represented by this Y-shaped structure. An enzyme is linked to it. And this is so that we can detect its presence with a color change. And that's where the sorbate part refers to, right? We're going to actually be able to create color, right? Which could be read as an absorbance, right, on a spectrophotometer. And then the assay is a series of steps. So what is an antibody? It's a product of your immune system. It's produced by a special type of white blood cells known as plasma cells, which are derived from B lymphocytes, commonly referred to just as B cells, right? They actually make antibodies that are specific to antigens. So what's an antigen? Those of you guys in lecture should know this already, right? The test was on it earlier, right? They're foreign substances that the immune system uses to respond, and they bind specifically to a specific antigen. And it's one of the most common responses of our immune system is to produce an antibody. So for this test, though, we need antibodies to bind to other antibodies. That secondary antibody is actually going to bind to the primary antibody. So that antibody is a special type of antibody, referred to as an anti-antibody, which is a mouthful, right? It's like, how many antis can you put in there, right? So sometimes what we do to make it not so anti, right, is we use one of the synonyms for an antibody, and that is immunoglobulin, right? Immuno, again, referring to it's a product of your immune system. Globulin is actually a protein, right? So we're going to use an immunoglobulin antibody, which means an antibody against an antibody. Still with me? I know. It's frightening. Okay. So those of you guys in lecture, right, where do the antigens bind to the antibody? A, B, or C? And B. And, right? So they like kind of grab onto it with their arms, you could say, right? And yeah, in the variable region, right? This section of the part of the protein that's known as the light chains, and then you have the heavy chains. This portion is variable, is different for each antibody. Now, so our antibody, our primary antibody, is going to grab onto the antigen with their arms. So now, where's the secondary antibody going to bind? It's C, right? At what we call the FC region, right? It's going to stick on there. So we're going to create an antibody sandwich today, right? We're going to go antigen, primary antibody, secondary antibody, right? We're going to stack them all up like you stack layers of a sandwich. So why are they different colors in this diagram? Are all antibodies the same? No, each antibody is different. So this is to represent that if we took a sample of serum from somebody's body, we're going to get all different antibodies, right? So, as we said, right, at the top is where it varies because that's where it binds to the antigen, okay? The bottom portion is what's consistent so the immune system can recognize it. And you actually make 5 different bottom portions. portions of your antibodies. There's 5 different classes of antibody, which you don't have to know for this class, but for lecture you did, right? And so we commonly abbreviated IG for immunoglobulin and then a letter for what class the antibody is. We're going to be working with all IgGs today, right? So all going to be this simple Y structure. We're not working with any human stuff today, right? So no gloves needed, no goggles, no aprons even. You could technically eat it if you really wanted to. But we're not going to eat anything in this lab because remember in this lab there are other things that would make you very sick like the stuff on the desk already. So the specific portion of the antigen that the antibody binds to is referred to as its epitope because you'll notice it only grabs onto a piece of the antigen, not the whole thing, right? Because it's a molecular interaction. So your antibodies can be found in the liquid phase of your blood. Depending on how that blood is taken from you, how that sample is taken, if they take it in an anticoagulant tube and then they spin out, centrifuge it, and all the red blood cells and the white blood cells are going to create a little coat, that liquid phase is referred to as plasma. As an immunologist, I was kind of snicker at my boss when he would give me plasma. And the reason why he would do that is because he only wanted to take 1 tube from the animal, right? So he'd run a whole bunch of tests, CBC count, right, all these things on that non-coagulated blood. And then they would spin it down and they would give it to me so I could pull off the plasma and test for the antibodies. That was because, again, they just wanted to take 1 tube from the animal. If we want to run serological tests where we're really looking for antibodies, we're going to do what's called a serum sample. We're going to take the blood in a tube that's going to allow it, actually promote it to clot. So what happens is all those clotting proteins get in the clot, right? And they're not in the liquid phase. Unlike plasma, because the blood didn't clot, guess where all those proteins are? In the liquid phase. And so they kind of gunk up these assays that we try to do because in addition to the antibodies, you get all these other proteins that are usually associated with clotting, right? And they can kind of get in the way sometimes. So I prefer when I have the choice, right? Please give me my serum tube, right? So that way, you know, I didn't have those clotting proteins, and I mostly just had the antibodies. And that's what I was interested in, right? How many antibodies does my animal have at this stage of infection with leprosy, right? How far along is this animal? Does that make sense to you guys? Okay. So serological testing, they're referring to, again, a serum sample, and they're usually looking for antibodies. So what are we doing today again? Yeah, antibody detection. We want to know, right, if your sample contains an antibody. But you can do this test and actually look to see if the person has an antigen. Okay? You just do the steps. Your sample is just a different step in the process. So Bio-Rad is a really great animation. This is, again, a kit from Bio-Rad, a company. And so I have their animation link right here. It does not have any audio. So don't be like trying to fix your speakers, right? Most things nowadays have audio, right? So it's like we're all like, why isn't it making any noise, right? So for us, which one are we clicking on? Yeah, we're doing antibody, right? So the first step is we're going to start with a strip of plastic wells that I'm going to give you guys. Each group is going to get a strip. Then you're going to add purified antigen into every single one of those wells. And we're going to let it sit for a little while, right, 5 minutes. Then we're going to dump it out onto some paper towels. And then we're going to put some wash buffer in. And what that's going to do is anything that didn't stick is going to get washed out. And it's also going to coat the areas where the antigen didn't stick to stop non-specific binding. So in your lab manual, it has a blocking step. Our wash buffer works as a good blocker, so we don't do the blocking step. So then we're going to add the primary antibodies. So again, there could be lots of different antibodies there, but we're only interested in one, the one that binds to our antigen, right? So when it binds, right, when we wash, it's going to stay stuck. The other guys that didn't bind are going to get washed away, right? So we'll wash 2 times, right? And we're going to remove anything that did not stick. Goodbye. What's the next step? Yeah, we need the enzyme now. So that's attached to the secondary antibody. Where is it going to bind? Is it going to bind to the antigen or the primary antibody? The primary antibody, right? If it bound to the antigen, why do we even need the primary antibody? Right? Well, because we're trying to find the primary, right? So we need the secondary antibody that has the enzyme linked to it, right? So the cute little Pac-Mans. And again, notice where they bind. They bind to the antibody, the primary antibody that's binding to the antigen, right? They're binding up here. And they have the little enzyme attached. So then the very last step, after we wash, and when we get to this point, we're going to wash 3 times because lots of stuff in there, right? We want to get everything that's not bound washed out. Then we're going to add our substrate, which is colorless. But when you add it, it interacts with the enzyme and it turns, wait for it, blue. Yay. Clear, blue, easy. What is that? Pregnancy test, right? Guess what you're doing when you do a pregnancy test? This on a filter paper. That's it. Right? What's the antigen? You're looking for the antigen in this case. What do pregnant women produce? Hormone. Human chorionic growth hormone. In excessive levels, so much so that it spills over to your urine. That's why you can pee on a stick. Right? It's that simple, right? Your hormone's the antigen. The test kit has a primary antibody just for that. Then a secondary to bind to that antibody if the antigen was present, right? You got antigen, you get antibody, enzyme, substrate, color change. Right? It's always those 4 things lined up. So that pregnancy test, as I said, right? You're starting out with all these possible things in that, right? So bunch of different things. Right? And some things are going to stick and other things aren't going to stick. And we're going to wash away whatever doesn't stick. And then we're going to apply the primary antibody that's going to bind to that antigen. In this case, human chorionic growth hormone, right? It's only going to bind to that. So a purified primary antibody sticks to it. Then, of course, we can't see this, right? So now we need our... It's going to bind to the primary antibody. Right, the secondary antibody. We'll keep it easy, right? With the enzyme on it, right? That's the key there, right? That good old enzyme. And then, so when we add the substrate, what happens? It turns blue. Same thing, it just depends on what's the test sample. Is it the antigen we're looking for, or is it the antibody? Right, today it's the antibody. So, if we were doing antigen detection, which is commonly referred to as the direct ELISA, right, we want to know, is the antigen there? So we're going to use a purified, commercially available antibody that's only going to bind to that antigen we're looking for, right? So what tests do this? Good old pregnancy test, rapid strep test, ovulation test, HIV antigen test, drugs, flu tests, right? All these rapid tests, that's what's happening, y'all. That's the mystery, right? If the antigen's there, the primary antibody's binding to it, then the secondary binds to the primary antibody, enzyme catalyzes reaction, color change happens. So, do we sometimes want to know if you've got an antibody? Yeah. Why would we want to know if you made an antibody? Those of you guys going into nursing school know about this, right? They want your antibody titers to prove you've been what? Vaccinated, right? But we can use it for disease detection as well, right? So again, we're asking the question here, what antibody do you have? So we have to start with a specific antigen, right? A purified antigen, so that your antibody only binds to that antigen, right? And then we put the secondary to be able to detect that primary antibody that's in your sample. So HIV antibodies, right, we can detect for that. A lot of times when they do HIV testing, they do both antigen and antibody testing. Heartworm. When you bring your dog to the vet and they take a blood sample, they're looking for antibodies against heartworm, right? Hepatitis viruses. And again, like right now, a lot of nurses, right, they're requiring you to get your titers. How many antibodies do you have against the things you should have been vaccinated against, right? They can do this with this specific test with the Alisa. But they'll do indirect, right, because they're looking for antibodies. So I'm going to skip over this HIV stuff. But, you know, you wouldn't want to tell somebody that they have HIV unless you were sure. So today, as I said, we're going to make an antibody sandwich, right? We're going to start with a an immobilized surface. And it could even be a slide. Doesn't necessarily for some of these tests. In the case of the pregnancy test, it's not done in a well, but on a filter paper strip, so that the reagents come in contact with stuff as it moves through the filter paper. Some tests that we do, we immobilize stuff onto a membrane, right, onto like a little plastic sheet. So the important thing is that we've got to have a stable surface, right, that we can stick stuff to, we can immobilize stuff to. So the first step is an antigen, right? We've got to apply an antigen or have an antigen in our sample that we're looking for. And then the primary antibody is going to bind specifically to that antigen, right? And then the secondary antibody binds to the primary antibody, right? It's an antibody against an antibody. So it only binds to the antibody, not the antigen, right? We're layering up. And this one, of course, is important because it has the enzyme conjugated to it. And when the substrate's added, a colorless substrate, it converts into a colored substrate, and so we get a color product. Some other enzymes can actually produce fluorescent products, and some can actually even be chemoluminescent products, right? It isn't always just a straight-up color change. It could be a fluorescent color change, where you would have to use what to be able to see the fluorescence? You'd have to use UV light. So here's an example, right, of cells on a slide, right? So this is a real research paper, right, real stuff, where they used antibodies specific to specific proteins that had targets, enzymes, right? And they would catalyze a substrate and they would fluoresce. So under UV light, they were able to see, right, where this particular antibody bound to this specific protein. You can see the nucleus gets stains blue, but you can see these proteins, where they're located in the cell. And they're kind of near the nucleus, huh? Where these guys are all spread out, this particular protein they were looking at. And this other one all spread out throughout the cell. And then this other protein, look, it was only around the edges of the cells, right? So probably associated with the membrane, huh? Right? So really pretty to look at, right? But very important, just like when we were talking about last time, you know, why would you want to make a sperm glow? So we could tell one from the other, right? So we can look under the microscope and see, where are those proteins, right? What are they doing maybe? Right? We can ask so many more questions when we can basically color stuff at the molecular level. Okay. So, for our procedure, we're going to label our plates, our tubes. So inside your box, you'll see a demo, right? Do you see that? So that's how we're going to label. You'll notice we're going to do everything in triplicate. So we're going to have positive and negative controls. You don't have your strips yet. I just wanted you guys to find the example. Okay. So what's our first step going to be? I'm going to give you a rack with little tubes. The green tube that has the plus on it, doesn't have anything on it. The green tube is the antigen, right? So it's the first one we're doing. And I didn't come up with these colors. It's in your manual in the kit. But kind of makes sense. Green, right? Go. That's our first one, right? So we're going to put 50 microliters in every single little well. We're going to let it sit for 5 minutes, right? Then we're going to dump it out, and I'm going to show you how to do that. And we're going to wash. We're going to add wash buffer, and I'll show you how to do that. We're going to do that once, dump it out, put the wash buffer again, dump it out, and really dump it out the last time, right, before we're going to add in our test samples. So let's go ahead and do that. Step, and I'm going to hit pause. So we just did this step, so we've got 5 minutes to kill, so let's talk about stuff. Right? So what you're going to do after our 5 minutes are up is, as I said, I'm going to show you how to dump this so you don't cross-contaminate your wells later on when the wells are different. Right now they're all the same, right? But good technique is important. So we're going to go through a lot of paper towels, right? We're just going to blot onto the paper towels, and I'll show you how to do that. Remember, we're going to wash, right, before we do the primary antibody. And then we're going to wash a second time, so we're going to do it twice. Why do we wash? Wash away what doesn't stick, right? Anything that's unbound we want to wash away. Okay, so what would be the consequences if we wash too much, though? Bye, Renisha. Yeah, and do we need stuff to stick for this test to work? Yeah, so if it doesn't stick, what kind of results are we going to get? Negative, but are they going to be truly negative? They're going to be false negatives, and we certainly don't want that, though, right? We don't want false results, right? So most or all, right, we could remove it and end up with false results. What if we don't wash enough, though? Yeah, it might get too crowded, right? So that when we add other stuff, the stuff that isn't bound binds to our other stuff, and we end up washing it away, right? So it wouldn't necessarily make our tests inaccurate as far as positive or negative. We will probably still get positive results if it is truly positive, but it won't be at the same level, right? Because some of our stuff would be removed by the stuff that wasn't bound. So this test can actually be done quantitatively, where we actually get a number. We could put it on a spectrophotometer and get a number for about how much color is produced. Would give us about a number about how many antibodies are present. For our purposes today, because I haven't adapted this lab yet for that, because we do have a plate reader next door now that can read plates, and a spectrophotometer can read a whole plate all at once. It's called a plate reader. We're just doing positive and negative today, right? But yeah, it would throw off, right, the accuracy or sensitivity of our test if we didn't wash properly. So again, we're skipping the blocking agent, right? Because some of the components in our wash buffer, and it's buffered so it's balanced for saline. It is a wash, so it's going to bubble. It's got a detergent in it, right? So we, again, want to make sure we get all the bubbles out before we add our next reagents. So we're going to add to the plus wells, the plus tube, our positive control tube. And what does this tube contain? What are we testing for today? Which antibody? The primary, right? The primary antibody that binds to the antigen. So that's what's in your little tubes that have the plus on it that are blue. Why do we do positive controls?
Sure, the experiment works.
Right. If you do the pregnancy test, y'all, 2 lines is pregnant. Why? One's your test sample, the other one is the control. If the control doesn't even work, the test tells you error, repeat, right? You want that control to work, right? And so it helps with the reliability of your results, right? The control works, chances are, yep, you're pregnant. Don't take another one, y'all. This is a highly sensitive test. This is a highly specific test and sensitive test. So if it is showing positive, you are pregnant. Don't do like Juno did and go get 6 more. They're all going to come out positive. Figure out what you're going to do about it being positive, right? Can you get a negative result, though, and still be pregnant? Yeah, because, again, it's really good in sensitivity, but, I mean, it's not going to detect really tiny amounts, right? So negative isn't sometimes a negative, right? It depends on the sensitivity of your test, right? So we want to make sure it's working properly. Negative controls, right? What's the purpose? What possible would cause negative results in your positive control? So we said, how could we end up with negative? Do too much what? Too much washing, right? Too much washing. You added the wrong reagents, right? Your reagents are expired, which isn't the case. These are fresh, new. No problems like the ampicillin. All right, so you guys want to learn the next step? Let's pause. Do the next. All right, so positive controls, right? What do they have in them? The antibody, right? The primary antibody that binds to the antigen, right? So it would look like this, right? Antigen, antibody, right? Negative controls, right? What's not going to be in the negative control? What do we leave out that makes it negative? Which antibody? Primary. No primary antibody, right? Why do we do that? Why have negative controls? How could we end up with a positive result and it not actually be what we're looking for? Could that happen? It shouldn't, right? But we want to make sure that just that antibody is what's going to cause that reaction, right? So, you can't get positive results without the primary antibody, right? We want to make sure that that's the only thing that's going to cause a positive result. Is that? Yeah, you could do it on water, right? Water should give you a positive pregnancy test. If it does, then we got some contaminated water, right? Exactly. Yeah, they always say it's the water, right? Okay, what are possible causes of positive results in all our negative wells? How can we get positives in our negative? Something went wrong, right? Somebody did something wrong, right? Maybe you forgot to wash, right? But, the negative control shouldn't have it in there, right? So that shouldn't be an issue. Contamination, again, this is why, especially on this next one, we want to tip sideways. We don't want to cross-contaminate well to well, although that wouldn't necessarily contaminate the whole thing. But definitely, you don't want to re-blot on your paper towel, right? You want to hit on different sections, so you don't pick up anything from the paper towels as well. How would positive result in all the negative wells affect your interpretations of your results? So if you do this test and all the negative wells come out positive, they're all blue, it's an invalid test, right? You can't trust that. What went wrong? You don't know, right? You wouldn't trust those results, right? And you certainly wouldn't want to tell a patient they have HIV, and in fact they do not, right? You want to be sure about this, right? So if your negative control doesn't come out right, you can't trust that test. What should they look like? Just antigen, right? The purified antigen we put in, there should be no primary antibody here. So then you have two patients that you're looking at today, 3 and 4. As you can guess, somebody's positive and somebody's negative. Your challenge today is to find out who, right? What the answer is. So we did all of this in triplicate. We do that just to waste time and reagents? Sure, why not, right? It's Delgado. We got money coming out our ears, right? Not hardly. Why would we do it 3 times, y'all? What's the likelihood you're going to screw it up 3 times? Hopefully slim, right? So the more often we test something, the more likely we're going to get an accurate result, right? And we want high accuracy and high precision, right? We want them all to be the same. If they're not, right, then we've got some problems that we need to address, right? And so, again, if you have inconsistencies, what might be the reason for that? Well, this is your first time ever doing it. You're going to probably do it perfectly? Yeah, why not, right? No. Right? There's a little bit of technique involved, right? Or it could be a poor sensitivity issue, right? Why you might get a little brighter color in one than the other, right? Variabilities in your replicates, right? If one of them, right, like say our positive control, 2 of them come out really bright blue and then the other one nothing. You're going to be like, oh yeah, it's fine. No, because they should all be exactly the same, right? If they are truly exactly the same, they should all be exactly the same. Still with me? All right. So our patients, as I said, 2 choices. Somebody's going to have antigen with the antibody we're looking for. They're going to be positive, right? And the other one is going to be negative. No antibody that we're looking for. Okay? This is a 96 well plate. I have one of them up on my desktop. This is what I would do when I worked in research with the samples that I was doing testing for antibodies. So these would be my test samples, and actually I did them in quadruplet when I did it. I did it 4 wells for each one of my samples. And then down the front of my well, of course, right, you have your negative control, and then I had my positive control, and I even did that in quadruplet. So on my plates, I would divide them up, right? This would be, actually I didn't usually do a negative control for this test. I did not. But I do 4 wells, all my control. But you see how their positive control, it's really light here and then gets darker? That's because, you know, it's more concentrated here and less and less and less, so therefore less color. Because like I said, if you put it on a spectrophotometer, you can get a number and know about how many antibodies are in these samples. So you can see these samples down here are really positive, right? And some up in here are positive. Some of these are probably negative because they're really light. Does that make sense? And again, for some of these tests, a number would be, you know, for me, we looked for 700 or higher was a good, strong positive. Less than that, we didn't consider it positive, right? And so I would get values like 1,000 when they were really far along in the progression of the disease of having leprosy. Okay, so we're going to wash, right, to remove anything that's not binding, right? And we're probably at the time where we could do that. We don't want to wash too much, right, because we'd remove too much stuff, right? And we'd end up with false negatives. And we certainly want to wash enough, though, right, that we don't leave stuff behind and it doesn't soak up our next reagent that we're going to add. So what we just did is add the secondary. Remember, where is it going to bind? To the primary antibody, right? What should our negative controls look like at this point? So what's the only thing that should be in the tube for a negative control? What do we start out with? Antigen. That's it. Because no primary antibody, right, for negative. So that's it. And so again, nothing for the secondary to bind to, right? Everyone still with me? All right. Okay. So for the positive wells, though, right, what's it binding to? And again, look right here, to the primary antibody. Secondary binds to primary. Secondary, primary. There's a reason why we call them that. One comes before the other. All right, so we're waiting 5 minutes, right? Our negative should be just antigen, right? Okay. This time, after our 5 minutes, you're going to wash how many times? 3 times. Again, we don't want to wash too much, right, because we'll wash stuff out, right? But we got to wash enough, and in this time, this is why we're washing 3 times, because especially for our positives, we got a lot of stuff in there now, right? We've got antigen, primary antibody, and now secondary antibody. They can kind of get caught up in each other. We want to make sure to get them all out, right? So again, we're not soaking up. And definitely for this one, right, if secondary is present, you're going to get a false positive, right? and our negative controls, we've got to make sure we wash out. Although, again, it shouldn't get hung up, right, because it's just antigen in those wells, and we know the secondary is not supposed to bind to the antigen, right? It's supposed to bind to the first antibody. Still with me? All right. So this is probably the most important wash step, right? And making sure you don't forget to do it. So 3 times, right? So before I give you your substrate, right, I'm going to say, did you wash 3 times, right? Because that's the last step, y'all. And the substrate is sensitive to light, so it's in the refrigerator wrapped in foil, right? So I'll give it to you when you're ready for it. And of course, it's only going to interact with, and we're not going to bother putting the foil over it. You can if you really want to, but I mean, the reaction is instantaneous, y'all. It like takes seconds once you put it in. So we'll be able to know our answer right away. Normally you would cover it up with foil, right? Waiting to use it in the plate reader, right, to keep it from overreacting. So you wouldn't want to wait more than 5 minutes before you would read it on the spectrophotometer. So I could only do so many plates at a time because I could only go so fast, right, as far as putting it on the machine and reading it and getting the next plate on and reading it as well. But sometimes what I would do too is I would wait to put the substrate in, right, so that I could run a set of plates, then do the last step, run a set of plates, right, so I don't have to worry about the time for that. So why is it important not to mix these reagents during the experiment or to let the solutions overflow with each other? Yeah, you're not going to get the correct result, right? If you just mix it all up, right? You're going to get false positive results. We don't want that. We want it to happen, right, on that immobilized surface, right? We want this series of reactions. We want the antigen stuck, right? Anything that doesn't stick, we want to wash away. We want the primary antibody that just binds to the antigen to stick. Anything that doesn't bind to get washed away. Then we want the secondary antibody that has the enzyme linked to it to bind to just the primary antibody. So when we add our substrate, again, wash anybody who doesn't stick, that we get our color change, right? Our antibody sandwich today, right? So lots of different techniques. Like I said, you employ this, right? So if you've heard of dot plots or Western blots, what they usually do is they spread out the proteins by gel electrophoresis first, then they transfer them to a membrane, and then they do exactly what we just did. They just stick the membrane in a little dish, right, that contains the different components. So, you know, you'd have your antigen, say, on your membrane, then you'd add antibody solution to that membrane, you'd shake it up in liquid, right, then you take the membrane out and you would dip it in different wash vessels, and then you'd stick it in the secondary antibody and then your substrate, and then you would read your results. And in this case, on the piece of paper, the membrane, you'd see the different lines where the different stuff that you're looking for. You know, so lots of cool stuff. Affinity chromography, where they can actually separate out things and identify things. And if you're interested in this cool type stuff, right, look into the biotech classes, right? If you think this is cool, you like doing this kind of stuff, maybe do some protein purifications and do tests like this to confirm you got the protein you were supposed to get, right? You can do cool things like that. So for our kit today, right, we have our microtiter well that we're using as an immobilizing surface. You're actually starting out with gamma globulin, right, so IgG from a chicken. So we actually, our antigen is an antibody we started with today, right? And we stuck that in our wells, and then they took that chicken antibody and they stuck it in a rabbit. So then a rabbit made an antibody against the chicken. So you have what's called rabbit anti-chicken antibodies, right? So guess what we're going to do next? Now we need another animal to make anti-rabbit antibodies, right? So now we have a goat, right, with anti-rabbit antibodies, right, IgGs, with the enzyme attached to it. And the enzyme is horseradish peroxidase. So it takes our substrate that contains, oh, look at an old friend. Who's that? Hydrogen peroxide, right? And this reagent, and when we oxidize the reagent, it becomes blue. So where is that? Yeah. So it does decompose, right? So we keep it at 4 degrees Celsius, and we keep it in the dark. So I got it covered up in foil so that this reaction doesn't happen, but because of the enzyme. So horseradish peroxidase enzyme converts this product. So colorless here, then the conversion makes it blue. You would read it using a special filter so that we could read the blue light intensity on the spectrophotometer, which is in the 250 nanometers range on the spectrophotometer. Oh yeah, just has to happen.